So thank you very much both for the very kind introduction, and also to Alison and Brian for inviting me to speak. So I'm going to start by saying I really feel quite a fraud being here. I think John said earlier on he was partial to an equation. And I should be honest that I'm really not made that way. And in fact, if you started getting me to try and interpret some of the equations that you've shown, I'd start shifting pretty uncomfortably. So what I do is, so my big interest is the tumor microenvironment in prostate and bladder cancer. So I spend lots of time looking at these rather beautiful multicolored fluorescent images. And so these are 5 bladder cancers, and I focus on a cell type called cancer-associated fibroblasts, or CAFs. And as was mentioned, I've sort of come through the academic route very slowly and arguably faffed around working abroad. But I now treat patients with prostate and bladder cancer using both drugs and radiotherapy. And I work with a wonderful lady called Prof. Ros Eels. And when I became a consultant at the Royal Marsden, she very generously asked me if I would co-author an editorial about the Armitage model. And I didn't directly reply saying, gosh, I don't know what that is at the time. I said, of course, I'd be delighted to co-author with you. And then started reading the paper. So what I'm going to do in this talk is very much come at the original Armitage model with the perspective of somebody with a biological clinical angle who is very biostatistically ignorant. Okay, so I'm going to start by summarizing what I saw as the 4 different patterns that were summarized in the original Armitage model, and then describe a contemporary reanalysis that we did and published in the British Journal of Cancer a couple of years ago, and then bring out some further biological considerations, because I was really fascinated by the insights surrounding some of the assumptions in the Armitage paper, so what they sort of meant to me as a biologist. So starting with the 4 patterns, so as was just mentioned by Anthony, the first sort of pattern that's described in the original Armitage model is this pattern, and in this case we see it with pancreatic cancer, where you're talking about a cancer with no known environmental or hereditable factors in etiology, where you see an increase in incidence approximately to the 6th power of age at a relatively constant rate. So we've got quite a nice straight line in both men and women with pancreatic cancer. We now switch to a second group or pattern that was seen, where you have a greater than expected incidence in the named cancer at a younger age. And in the original BJC paper, then Armitage and Dole describe the potential role of heritable cancer syndromes. And you can hopefully see this increase here in both men and women. So I think at that time, St. Mark's had a registry of patients and families with familial adenomatous polyposis. But our understanding of heritable colorectal cancer syndromes was much less than it is now. And in fact, you know, in the 1980s, Henry Lynch and colleagues were still trying to persuade the global community with much skepticism that hereditary colorectal cancer was something that we had to take seriously. So I now treat quite a lot of patients with Lynch syndrome, and we're very familiar with it. But when Armitage and Dole published their paper, Lynch syndrome really wasn't at all known about. So the third pattern is particularly clear in lung cancer, and you can see that there's a reduced incidence of lung cancer in the older age groups in both men and women. And I think what I'd say was it was a very different era back then. And I came across these ads when I was preparing the talk, and I don't know whether the relevant physicians actually consented to these advertisements or whether they really believed what they were saying. But clearly the messages that doctors were giving patients about smoking was very different back then. But clearly, so it seems, the elder generations had the sense not to follow the fashions and fads of that day and smoked much less. And that's the explanation that's given for the reduced incidence in lung cancer in older age groups. So then the fourth and final group I'll describe is the cancers with endocrine, or we think endocrine drivers. And this is both breast and prostate cancer, where you have a real departure from the straight line. So the straight line is to the 6th power of the age, due to the influence of endocrine secretions, which they declare quite clearly vary throughout the course of a lifetime. Okay, so these are the 4 patterns that we went on to re-examine. Before I move on to that, one of the things that really struck me was how clearly Armitage and Dole said, you know, it will be assumed that the changes, whatever these changes are, must proceed in a unique order. And the time when the strength of factors responsible for the individual changes is of greatest importance varies according to which sequence and which change in the series is affected. So they really beautifully describe what we now know as the Vogelstein model of colorectal tumorigenesis, and it has been sort of shown to apply to many other cancer types, where you have a series of quite specific and distinct genetic changes that drive a series of pre-malignant and then overtly malignant changes. And I was fascinated that the wording they used summed up the Vogelstein model so beautifully. So then how do we approach a contemporary day sort of reanalysis of the Armitage-Dole model? So one of the big changes, obviously, is that incidence no longer equates with mortality. So back in the 1940s, they could, you know, sadly say, well, incidence is an equivalent to mortality, but fortunately, you know, treatment of cancer has progressed hugely. And so actually, we decided that we needed to model both incidence and mortality separately. So as well as that, you also need a friend who is very good with IT. So Richard Corbett said to me, oh, we can actually, I can help you extract the data from the original paper and help you extract data from modern day cancer registries in order to actually approach a contemporary day reanalysis. And in the interest of transparency, I should say that I have many things in common with Richard Corbett, including we parent 2 children. He's actually my husband. And you can see the edge of one child in the photo. So we've prepared a series of these plots that were published a couple of years ago. And so you can hopefully see the red line in these plots is the change with incidence to the 6th power of age, as was originally proposed by Nording. And it's in the original Armitage-Dole model. And so the Armitage-Dole data back in 1950 to 51 is shown in red. The 2018 incidence is shown in blue, and the 2018 mortality is shown in green. And if you look just with a wide-angled lens, what's really clear is how the blue dots are so separate from the green and the red dots, which I suppose, you know, really does show how much of an increase of incidence of breast and prostate cancer we've had. And you could speculate on a number of reasons why. It could include, you know, screening, the advent of prostate-specific antigen, ascertainment bias, and possibly a true increase in incidence. But when we looked at the data, that's one of the things initially that really struck us. And I'll come back to these curves in a minute. So starting with group 1, to me, the 3 curves here for pancreatic cancer in men are pretty much directly superimposed upon each other. And I think we still don't have knowledge of heritable or environmental risk factors for pancreatic cancer. And sadly, we still have very, very few effective treatments, which is why the incidence and mortality really, and particularly the mortality, hasn't changed in the last 60 years, sadly. So then we come to the second group I described, which is, as I show here, men with colon cancer, where although the dots are not directly on top of each other, there's a very similar trend where you see a similar pattern of increased incidence and sadly mortality at younger ages with probably the same hereditary factors that were described by Armitage and Dole themselves. So then you come to the lung cancer group, and I'm just showing the data for women here. And what you can see is that the red dots, particularly right up here, are really quite separate. And I think my explanation for this is that now smoking is very similar rates across the decades. So this idea that elderly patients were protected from smoking-induced lung cancer back in the 1950s because they didn't follow the fashions and fads of that day is now something of the past. So I think these changes are very in keeping with what Armitage and Dole themselves described. But what you might notice is that the green dot here, which is the mortality, has now shifted quite far away from the data back in the 1950s. And when I first saw this, it really took me by surprise. So I went to the Cancer Research UK mortality statistics for women. And I think you can explain the pattern of the green dot, so the mortality in 2018, by the mortality changes that have happened, which seem to be age specific. So the mortality changes have reduced a lot in the younger age groups, but continue to increase in the older age groups. So there's a sort of age specific mortality changes here. So then coming back to the endocrine-driven tumours, what really struck us with prostate cancer was the steepness of the gradient that you see in really all three lines. So the steepness here, if you compare the blue, the red and the green dots, it's much steeper than the red straight line of 6. And in fact, it's much steeper than all the other cancers. So what is it about prostate cancer that means that you have this particularly steep gradient? And the gradient is as high as 11, whereas the gradient for the other cancers was much more like sort of 5 to 6. So this is speculation, but a couple of views. So you will all be well aware that, you know, puberty brings a massive 20-fold surge in testosterone. And, you know, prostate cancer cells are directly driven by testosterone in terms of proliferation and growth. And I prescribe androgen deprivation therapy extremely commonly. And resistance to androgen deprivation therapy is often driven by massive amplification of the androgen receptor. So testosterone signaling is a very important part of prostate cancer carcinogenesis. But it may be that there's other factors involved in that steep gradient. And one big area of growth in prostate cancer is the growing recognition of chronic inflammation and potentially the interaction between certain microbial species and chronic inflammation in driving prostate cancer carcinogenesis. And what is emerging, and what I'm saying here is partly speculative, but there's more and more data coming out that specific microbial communities do appear to associate, and there's a suggestion that they're functionally related to specific gene fusions that we see in prostate cancer. So one idea I have for why the gradient is so steep in prostate cancer is there may be specific interactions between the testosterone surge in puberty and subsequent changes exerted by microbial communities. So now I'm going to move to a few assumptions and the sort of wider description of the model that Armitage and Dole gave in the paper that really caught my eye. And as Anthony mentioned, they emphasized the long latent period observed after exposure to a carcinogen as being sort of inherent to their model. And they describe in the paper quite a lot about squamous cell carcinoma of the penis and how that relates to circumcision and timing. And they say quite clearly, some change must take place within the first 14 years of life, which eventually leads to the development of the disease after a latent period of as long as 70 years. And I think if I'd been told that 10 or 15 years ago, I might have, if I'm honest, been quite skeptical. But the modern sort of genomic approach to cancer has really shown in some cancers how accurate they were. And this is one figure from the recent TracerX renal study led by the Swanton Lab and Samra Tarajlik. And it was all over the Daily Mail, this, you know, this amazing, you know, big groundbreaking change that actually some of the initial cancer-driving mutations occurred in adolescence, yet actually it had been described very clearly, you know, 60, 70 years before by Armitage and Dole. And I think the genomic studies that are emerging where you're timing pre-malignant changes are very consistent with the TracerX renal data. I was really fascinated that, you know, the BJC paper clearly describes a complex process of 6 or 7 stages. And again, when you look at the data from genomic sequencing, then you're often left with trying to understand what 300 or 400 different mutations might be doing and what a whole series of different copy number alterations may mean. And the data is, bewilderingly complex, and you really need a bioinformatician to help you out to understand what on earth might be happening. But if you look at a recent pan-cancer analysis of whole genomes, so this is a massive international consortium effort where they looked at the whole cancer genome data from over 2,500 genomes across 38 tumor types. And what I show here is a bit of text from the abstract, where when you actually look at driver events and you filter out, and this is done by people who understand filtering out passenger mutations versus driver mutations much more clearly, then they suggested that 4 to 5 driver mutations on average were present in individual tumors, and that this was likely to be an underestimation of the number of driver events. So actually, in my opinion, that's very close to 6 or 7 stages. Until quite recently, we very much thought that individual mutations were the drivers. But what's now emerging is a single discrete event can occur, be it chromosome shattering or different chromosomal catastrophic events that can affect a huge number of genetic loci and involve a huge number of mutations, but they are a discrete event. So the final thing that sort of really caught my eye was the statement in the BJC paper that successive cellular changes are not necessarily mutations. So we had a conference earlier this year at the CRIC that was called Revisiting Berenbloom. And Armitage and Doll go on to kind of write in the paper that the evidence exists that carcinogenic and mutagenic activity are not always identical. And I think we are seeing with fantastic data coming, really from Cambridge and elsewhere, that what we assumed was a direct effect of carcinogens causing mutations is not always being borne out as clearly as we'd thought. And my understanding from some of the CRUK Cancer Grand Challenge projects that are going on is the assumptions that specific carcinogens work through mutations are being called into question. And there was a really nice paper that came out last year from Charlie Swanton's group that gives one example of this and relates to the non-smoking associated lung cancer that you talked about, Anthony. So this paper describes a mechanism of carcinogenesis of EGFR-associated lung adenocarcinoma, which is a typically non-smoking associated lung cancer that occurs more commonly in women. What Charlie Swanton's group did was a combination of epidemiological analysis and use of functional mouse models. And what they showed epidemiologically is that there was a tight association between global areas where there was high amounts of particulate matter, which is what this PM refers to, and the incidence of EGFR amplified lung adenocarcinoma. And having seen that epidemiologically, they went on to explore mechanisms in a mouse model where they induced EGFR mutation using a Cre recombinase system. And they went on to show that when you added particulate matter to the mouse environment, then the number of lung cancer lesions increased substantially. And you can see the control lung and then a lung where the mouse was exposed to particulate matter. So a striking increase in the development of lung tumours. They then went on to look at the lungs of several hundred patients in different cohorts who did not have lung cancer. And what they showed was association with age and the presence of EGFR mutations in healthy lungs that did not have cancer. So this goes back to the idea of there are lesions with oncogenic mutations, or there are areas with oncogenic mutations that do not become cancerous. So the bottom line in terms of the mechanistic insights was they showed that in the lungs of mice that were exposed to the particulate matter, there was an increase in a specific type of macrophage that was producing interleukin-1 beta. So I'm summarizing a huge amount of data, slightly simplistic here, but when they functionally blocked that interleukin-1 beta production with an anti-IL-1 beta here, then there was a dramatic reduction in the number of tumors. And this paper fascinates me because it's a really clear example of a carcinogen working via a non-mutagenic process. And again, when I read the Armitage-Dole suggestions that successive cellular changes are not necessarily mutations, I thought it was amazing that they could say that sort of 70 years ago. And you know, we're realizing how right they were in saying that today. So to summarize, I find the Armitage-Dole approach to epidemiological modeling of cancer really fascinating. And I think a lot of what they said in their paper is still relevant to a contemporary day reanalysis. And coming at reading the model from a non-mathematical perspective, I was fascinated in sort of the biological insights and the way they describe the assumptions and parameters of the model. So I'd like to thank Roz and Richard very much for a lot of help with the reanalysis we did, as well as my research group at the ICR, and Nick, Alan, and Kevin, who are my big mentors. And thank you very much to all of you for listening.